Okay, this is April 13th, practice speech here at the negative side. Question is, should gain-of-function research on potential pandemic pathogens be prohibited? So Akshaya Arjunan is on the opposition side, so this is going to be saying no. So first time all the way through, recorded. Good luck.
Thank you. I'd first like to take the time to thank everyone and the Honors College for allowing me the opportunity to participate in this debate. I think it's really important, especially as a researcher myself and for the research world in general, for us to discuss this topic and bring more discussion about the potential benefits and risks of this type of research. So on October of this past year, the U.S. government made a surprising move in the scientific world when it announced that it would temporarily stop funding new research that makes certain viruses more deadly or transmissible. Moreover, the White House Office of Science and Technology Policy asked researchers who conduct gain-of-function research on influenza or severe acute respiratory syndrome and Middle East respiratory syndrome to stop their work until a risk assessment is completed. The surprising move has led to much confusion among researchers due to the ambiguity and practicality of stopping funds for such research. Dr. Imperial and I stand in firm opposition of the resolution that gain-of-function research should be prohibited for several key reasons. However, before proceeding with the debate, it is pertinent to define few key words or phrases in order to properly understand the concepts of this debate. According to the Office of the Assistant Secretary for Preparedness and Response, gain-of-function research refers to any modification of a biological agent that confers new or enhanced activity. For the purposes of this debate, we will be focusing on biological agents that are potential pandemic pathogens, PPP, or more specifically influenza. It is important to acknowledge that currently there have been no known research labs that have successfully conducted a gain-of-function research project that has led to increased transmissibility between two human subjects. However, because of the new finding of a research lab that conferred increased transmissibility between two ferrets, this type of research has come under question. Since both sides can agree that there are benefits and risks to this type of research, we must analyze this issue through a cost-benefit analysis. We argue that while there may be potential risks to this type of research, the benefits far outweigh the costs, and prohibiting gain-of-function studies would severely limit the progress of basic science research. We stand in firm opposition based on three main points. Gain-of-function research is essential for a variety of research necessities. Biosafety measures can and are already implemented, and hindering progress in research provides more detriment than benefit. So first, gain-of-function research is essential for a variety of research necessities. Gain-of-function research is typically conducted in order to examine the impact that mutations or altered genes can have on the property or trait of an organism. It allows us to understand the fundamental nature of host-pathogen interactions and can shed light on the evolution of viruses and other infectious agents to acquire certain properties. Moreover, it may allow us to enable assessment of pandemic potential of emerging infectious agents, which could help inform public health and preparedness efforts, including developing effective medical countermeasures. Being able to understand how modifications in a genome can impact the transmissibility of a disease is undoubtedly a huge benefit to public health safety, especially in the context of creating vaccinations. The necessity of gain-of-function research can be seen in three categories: surveillance and diagnostics, virology and vaccine development, and the lack of alternative approaches. So first, let's look at surveillance and vaccine development. The influenza virus is one of a handful of infectious disease agents that can cause devastating pandemics with high mortality and morbidity in human populations. Given the biological characteristics of the influenza virus that ensure the continuous generation of antigenic variability, meaning that it mutates rapidly over time, this virus possesses a continuous extant threat, with the likelihood of new pandemics being determined by variables that are still poorly understood. In this environment, the influenza virus research community is humanity's best defense against the influenza virus. Consequently, anything that impacts influenza virus research, such as gain-of-function studies, is of utmost importance to public health research and well-being. It is important to acknowledge how molecular markers identified by GOF studies have provided important information for public health risk assessment of naturally occurring viruses. Moreover, GOF studies influence pandemic risk assessments through several key methods that make this type of research invaluable. According to a recent article by Cox Lab looking at the use of highly pathogenic avian influenza A in gain-of-function studies, there are numerous benefits for molecular-based surveillance and pandemic preparedness due to gain-of-function research. As soon as the H7N9 influenza sequence data was posted, the CDC and several other investigators identified several of the mutations to possess GOF phenotypes, such as increased respiratory transmission in ferrets, mammalian host adaptation, and enhanced virulence in mice. Early detection of these molecular markers in H7N9 viruses, isolated from humans, gave public health authorities evidence that these viruses pose an immediate pandemic threat, something that was accomplished only because of the gain-of-function research previously conducted. Based on H7N9 sequence data alone, development of a candidate vaccine virus began within a day. The viruses obtained from human cases in China were subsequently shared with international partners within 2 weeks so that additional virological characterization could be performed. Over a period of about 8 to 10 weeks, initial reports of human infection, numerous laboratories identified H7N9 viruses with increased binding affinity, replication in mouse and ferret models, limited transmission in ferrets, and reduced susceptibility to current antiviral drugs. These were all inferences obtained from molecular-based surveillance using knowledge obtained from GOF studies. As this data accumulated, the risk posed by H7N9 viruses compared to other influenza viruses was greater than for the currently known influenza subtype viruses. These collective findings, along with molecular markers that were identified, led to the World Health Organization collaborating centers for influenza and vaccine manufacturers to rapidly develop candidate vaccine viruses, and then also led the Department of Health and Human Services to perform human clinical trials to then stockpile H7N9 vaccines in the United States for pandemic preparedness. This shaved off months of time required to deploy this vaccine should it be needed in the future. Again, something that could have only been done with gain-of-function research. In recent years, both the range and speed of molecular surveillance for H5N1, another subtype of influenza, and other avian influenza viruses have continued to improve. Gain-of-function studies are needed to inform our interpretation of genetic data obtained from naturally occurring viruses. More data are required to fully elucidate the mechanisms by which influenza viruses with pandemic potential could cause severe disease and how they evolve during replication. In addition, when we look at alternative approaches, we see that gain-of-function research provides us a unique ability to prove causality, whereas other types of alternative approaches cannot prove this. For example, loss-of-function approaches will show that modification or deletion of almost every gene of a pathogen could result in reduction of replication or transmission. However, it doesn't show us a causality between a specific genotype and a phenotype. Bioinformatics and modeling approaches may be used to identify associations between a genotypic and phenotypic trait, but will rarely prove causality, since these are systems modeling and is using predictions to kind of show us what could happen in the future, but not necessarily showing a specific genotype to phenotype causality production. In vitro experiments on genes in isolation and studies with attenuated strains may identify causal relationships, but many phenotypes can only be investigated in the context of a wild-type pathogen, which is very hard to obtain. The next major point is that biosafety measures can and already are implemented. For example, there are already ways to build in safety features, such as the incorporation of a microRNA target sequence into the influenza virus genome that results in the inhibition of replication outside of the laboratory setting. So even if there was a chance of human error to occur that results in a virus being released into the air and potentially infecting a human, with the implementation of these types of safety features in the genome, we could prevent this released virus from being able to be transmitted between two humans. In addition, there are multiple layers designed in biocontainment measures, such that if one layer fails, others exist to mitigate the risks. For example, in a study conducted between 2004 and 2010, it analyzed the amount of laboratory-acquired infections that could occur due to human error. However, in none of these LAIs or laboratory-acquired infections that occurred, none of them involved viruses. Thus, it concluded that the risks of LAIs associated with viral pathogens should be estimated as less than 1 per 2,044. In addition, when we look at this hypothetically, if we were to limit doing this type of research just on the possibility that it could cause, some kind of a release of virus, what other things are we going to stop letting other people do? There are so many things that individuals in today's world are allowed to do, yet it could cause some kind of pandemic potentials. If we look at the case of just being able to drive, for example, how did we set the age limit that individuals are able to drive? Knowing the neurological capability of 16-year-olds versus 20-year-olds, what's to say that, increasing the age marker would reduce the potential of one person getting into a car accident versus a person of a higher age? So when we look at these hypothetical situations, you have to understand that when we're looking at the benefits of gain of function research, you are getting more benefits than costs, especially when we have biosafety measures in place to help mitigate any risks that might occur. For these reasons and more, my partner and I stand in firm opposition that gain of function research should be prohibited in the basic science research world, because when we look at it in a cost-benefit analysis, we see that the benefits much outweigh the risks. And for that reason, we stand in opposition. It's a lot longer than...
Oh my gosh, 11:20.